Welcome. We have been talking about discrimination measures based on spike train distances, and all these approaches that we have been talking about so far, starting from the stimulus reconstruction up to the discrimination approach based decoding, we have been talking about the case of single neurons. So it's actually not always true that based on the decoding is being performed based on single neurons. Actually, we always have a population of neurons based on which the entire population is projecting somewhere downstream, where the decoding is being done based on all this activity. And so it is important to note that the measures that we have used can be extended to a population of responses, whatever be the response measure. So for example, if we start with our response rate, rate as a response, so say we simply convert this rate into a vector where we have essentially, let us say, capital N elements where it represents the rate or rate response of the capital N neurons. Based on the entire population, we can develop all the measures that we have developed so far. If we want to decode the stimulus based on these rates, we can do the same thing, basically extend it with on the basis of the population or the vector R. So similarly, for the discrimination approaches, if we want, we can start with the KL distance approach, which is the best case for the all information based discrimination. There also, if you remember, we were looking at the KL distance between two distributions where we have P of R and Q of R, in response to two different stimuli, and this is our DKL. So this is in response to one stimulus, stimulus 1, and this is in response to stimulus 2. So we are getting two different distributions. So it is essentially, we convert this DKL into the DKL between two vectors, the distribution of vector random variables PR here and QR. So the joint distribution given a stimulus or for each and every stimulus that we are considering, we need the joint distribution of the responses or the rates or of the different stimuli. So once we have the joint distribution, the rest will follow exactly in the same way as we have discussed with our single neuron case or single response case. So with this in mind, we will now introduce you to the idea of how the correlations and the correlations in the variability of these responses become important. So far we have been, when we talk about the joint distribution, we are talking about the dependence between the response and the stimulus, where the response is a random vector where we have dependence between the different neurons as well as dependence in the responses of the different neurons. However, in this case, we are not considering another very important factor, which is how the responses are varying over different trials, and that if there are correlations in that variation, then also it becomes important in terms of decoding. in a population. And in fact, later on when we consider population based decoding examples, one particular case we will see how important the correlations in the variation are. So, if we look into that, let us say for a let us consider first two neurons whose neuron 1 and neuron 2, and let us say we consider a stimulus, a single stimulus, let us say S1. We will say S for now. Later on when we introduce another stimulus, that will be S2, and this one will become S1. So, let us say the response to the stimulus 1 of neuron 1 is R11, and response to stimulus 1 to neuron 2 is R12. So, if we now represent the response of the neuron 1, that is the, let us say, rate based responses along one axis, response rates, and the responses to the other neuron along the other axis, that is the response rate to neuron 2. So, for this stimulus S1 or S, we have the response R1 and R2 as the mean response estimated from, we are estimating this, as we have discussed multiple times, estimated from different or multiple repetitions, let us say m repetitions of the stimulus. So, this, let us say, this mean response R11 is here, and the mean response R12 is here. And so, we represent the mean response as a response that represents this particular point in this plane. And now, if we were to consider the different or the capital M repetitions of the responses, that is, they would be and represent each trial, each of those M repetitions as each trial, and represent that on the same plane, they would be scattered around this particular mean value of R11 and R12. So, let us say that they will be scattered in this manner around R11 and R12. So, this is in red is the mean, and in white dots are the different repetitions. There are capital M dots, and each dot represents how the response of one neuron is related to the other neuron for that trial. So, overall, if we look at this cloud of points around the red dot, which is the mean response, then we get an idea of how the two neurons' responses vary with each other over different trials. So, if let us say that how the responses vary between trials and is unrelated in the sense that the neuron 1 and neuron 2 responses do not relate to each other in any way, that is, they are varying independent of each other, then this same plot would look like this. So, if we have the red dot here, then we will see that it will be a circular kind of distribution of scattered points, that is, they are equally varied around the two response directions. That is, if N1 increases beyond its mean, that is, if the response of neuron 1 increases beyond its mean, we have a propensity to see all kinds of responses in N2. That is, all we are seeing N2 being larger than the mean response of N2 as well as lower than the mean response of N2. Similarly, when we have a decrease in response of N1 from the mean, then in different trials we again see that N2 varies equally above and below the mean response of the neuron 2. That means the change or noise, if you will, of in the response or the variation in the response around the mean of neuron 1 is not related to the variation of the response in neuron 2 around its mean. So, if we look at the correlation coefficient of these scatter of these dots or the pairs of responses of neuron 1 and neuron 2, in such a case the correlation would be near 0 or almost not significantly different from 0 statistically. However, let us say if there is a tendency of neuron 1 to respond of neuron 1 and neuron 2 to co-vary, that is they vary together around the mean, then when the neuron 1's response increases beyond its mean, there is a higher chance of neuron 2's response to be also above the response 2's mean. Similarly, if we, so this is our mean and so it is above. Similarly, when neuron 1's response is below its mean, then also we see a higher chance of neuron 2's response to be also below its mean. So that means their variation is correlated. That is, the responses from trial to trial between two neurons is correlated in a manner that is the fluctuations that we are seeing around the responses or around the mean responses of each of the neurons, they are related to each other in a positive correlation manner. So, if we look at the correlation coefficient in the responses of the pairs of responses in this scenario, the first case that we were drawing, we will see that the correlation coefficient rho is going to be greater than 0, significantly greater than 0, or it is positive. Here our rho is effectively 0, and so they are not correlated, and similarly now you can think of the remaining case where we have negative rho, that is, if the mean response again is here R11 and R12, then if we have the scatter around that red to be elliptical but sloping downwards on towards the right. So, this is our N1 response and this is our N2 response. We have a tendency of opposite variations around the mean of N1 and N2. And so, if we look at the correlation coefficient of these pairs of data, then that is going to be negative. So, this rho here, what we are calculating based on the pairs of responses in each trial, is what we call the noise correlations. So, noise meaning that the responses variation around the mean that we are referring to as noise. It is not necessarily noise as in not useful noise, not useful part of a signal, but just means the variation which we otherwise would have treated as noise. So, if we consider only the mean. So, now why is this important? Obviously, we will, as mentioned, we will see this in an example later on also. However, here if we think of it in the same way again, there is a pair of neurons N1 and N2, whose rates are being represented along the two axes. So this is N1, N2, we had stimulus 1, we had response R11 and R12. Now let us say we have stimulus 2, R21 and R22 are the mean responses again from M trials for the two stimuli S1 and S2. Now, as before, we will mark our R11 and R12 here. So this is our R11 and this is our R12. And let us say we have a scatter with positive noise correlations in this direction. And now if our, let us say, stimulus 2 mean we are representing in R21 R21 here, and R22 here, and we again have a distribution of points that is along this, and let us say the noise correlations are positive in both cases. Now, if you think about it, it is the positive noise correlations that is here is important in the sense that now let us say we, based on these responses, so let us say let me make it with the green one here. This is the overall spread of responses to the S2, and let us say this is the spread of responses to stimulus S1. So depending on how strongly correlated the responses of S1 and S2 are in terms of I shouldn't say responses of S1 and S2 are, it is the how strongly correlated the variation of responses are, that will determine how much overlap we have in this particular region. So this region, this overlapping region, is where the confusion region is. That is, response pairs of N1 and N2 within this overlapping region is going to be difficult to decipher as to whether it came from stimulus 1 or whether it came from stimulus 2, because we are seeing a finite or good probability of such responses from stimulus 1 as well as stimulus 2. And so if the noise correlations were actually lower than what they are, in that case we will find that if let us say somehow the noise correlations were actually lower, which means they would become more circular compared to the oval. Let me draw it in a separate drawing here. This is our R11 and R12, and this is our R21 and R22. So R21, R11, R12, R22, neuron 2, neuron 1. And now let us say the noise correlations were lower, which means that they become more circular than elliptical as above. And in the green also we will be seeing that it is circular, more circular compared to elliptical, and there will be a decrease in this overlapping region. So there, I mean, it's probabilistic, so there will be always some overlap, but the extent of overlap is going to be much lower if the noise correlations were lower. So this is the case where S1 and S2 responses are such that there is a positive correlation in the responses of the stimuli. So the correlation, now we are talking of a different correlation now, that is response of neuron 1 and neuron 2 in response to stimulus 1 and stimulus 2. Their responses change with a positive correlation. That is, if we take the mean, that is the red dot and the green dot, the mean responses are increasing with increasing rate of N1. There is an increase in rate of N2 when the stimulus is being varied. That is called signal correlations, because it is the mean that is being correlated here. So that is devoid of the noise, and hence it is as if it is signal and signal correlations. So for positive signal correlations in such a scenario is going to be that if there is a drop in noise correlation, then we will have a improved decodability of the stimuli. That is based on the pairs of responses, the degree of overlap between the response pairs is going to be reduced. So this is the case where the response pairs, mean response pairs, are positively correlated. Now if you go to the other scenario, let us say, where we have, let us say, our S1 was here and our S2 response mean is here, and now our spread of responses around the first stimulus and the spread of responses around the second stimulus is like so, which means that now we have a negative signal correlation. That is, the means are changing in a mean responses are changing in an opposite manner between neuron 1 and neuron 2. So now this is, but we have rho as greater than 0. We have positive signal correlations, and that is why we have the elliptical spread of response pairs around the mean, both in the green and the red case, S1 and S2. Now let us imagine that the noise correlations again are being reduced. That is, our rho is going down, and let us say from a positive value it can go on to, let us say if it could go on to a negative value, or even if it is positive and it is reduced. So the spread will reduce in this direction and will be made up by an expansion in the orthogonal direction. And so with the reduction in noise correlations, in this case, let us replicate this. Here we have the red elliptical spread and the green elliptical spread. I'm sorry. So after the change in the noise correlation, let us say, or if the noise correlations were less positive, we would still have an ellipse, but it would become wider in the orthogonal direction of change, or along the negative slope direction. So in other words, we are now getting an increased overlap as opposed to much low overlap. So, and hence the decodability of responses of neuron 1 and neuron 2 as a pair is reducing, because now we have more confusion in terms of decoding S1 and S2 or finding out which stimulus was played based on pairs of responses N1 and N2. So, now you can imagine, depending on how the signal correlations and noise correlations are, their combination can play a significant role in terms of decodability of responses, or finding out what stimulus was being played based on the responses. So, what we learn here, that even with pairs of neurons, based on the simultaneous changes in responses of the pairs of neurons in trial to trial. So, we are now considering the trial to trial variability. So, this trial to trial variability can profoundly affect the decodability. So, now this whole thing can be extended to a population of neurons in a higher dimensional space, and you can imagine how different combinations of signal and noise correlations and multiple stimuli in that case may influence decoding based on even just rate responses. So, how the responses vary over time also plays a role in decoding in the case of a population of neurons. That is, the relationship of this variation is extremely important. So, we will be revisiting this in an example later on, and now we will, in the next lecture from the next module, we will be starting with the statistical approaches in decoding based on responses, and we will be taking up examples as well from there on. Thank you.